Hello everyone, I'm Khalique Newaz from the University of Notre Dame. Today I'll discuss our work on network analysis of synonymous codon usage. We all know that there are four types of nucleotides. A combination of any three nucleotides gives a codon, which ultimately encodes an amino acid. Given the four types of nucleotides, there can be 64 different three nucleotide combinations, and thus there are 64 different codons. However, there are only 20 different amino acids. This is because many amino acids are encoded by more than one codon. All of the codons that encode the same amino acid are called synonymous codons. For example, the amino acid phenylalanine or PHE is encoded by two synonymous codons, TTT and TTC. Depending on species, some synonymous codons are used significantly less often than others, which makes them rare codons. For example, suppose that the amino acid phenylalanine occurs 100 times in a given sequence. Out of these 100 times, it is encoded by the codon TTC only 4 times, while it is encoded by the other codon 96 times. Here, TTC is the rarely used codon, while TTT is the commonly used codon. Please note that this is just an illustration, and there are more sophisticated mathematical models that are used to determine whether a codon is common or rare. It has been argued that rare codons can coordinate co-translational folding of the polypeptide chain by slowing down the translation process. Because of this, and because folds of orthologous proteins are often conserved, recently our collaborators at the University of Notre Dame argued that sequence positions of rare codons among orthologous proteins should also be conserved. They found that in multiple sequence alignment of orthologous proteins, there are a number of rare codon positions that are often conserved, suggesting that these conserved rare codons are a result of positive selection and are potentially related to conserved biological functions, including protein folding. In our study, we aim to further deepen insights into the role of synonymous codon usage in protein folding. Our hypothesis is that synonymous codon usage is linked to the final protein 3D structure and thus possibly to co-translational protein folding. Specifically, we examine whether amino acids encoded by conserved rare, non-conserved rare, and common codons occupy different positions in the 3D structure of a protein. We start our analysis with around 280,000 protein sequences for which we have codon usage information. Because we aim to study 3D structures of proteins, we focus on those protein sequences that have 3D data in the protein data bank. Additionally, we use several criteria to ensure as high quality data as possible, resulting in 63 proteins. To rank the importance of an amino acid in a protein 3D structure, we model protein 3D structures as protein structure networks. In a protein structure network, nodes are amino acids, and two nodes are joined by an edge if the corresponding amino acids are close enough in the 3D structure of the protein. Note that in our previous work, we have already shown that protein structure networks can correctly capture the 3D structures of proteins. We examine centralities of nodes in a protein structure network with the idea that centralities may be able to intuitively capture positions of amino acids in the protein 3D structure. Given a centrality measure, the higher the centrality value of a node, the more network central the given node is. An example of a centrality measure is the degree centrality, which measures the number of direct neighbors of a node. In the example on the right, since node B has a higher degree centrality than node A, node B is more network central than node A. In this work, given a protein structure network, we use 6 different centrality measures to study 3D structural positions of the conserved rare, non-conserved rare, and common codons. We verify that centralities correctly capture as central those amino acids that are known to be in the protein core and as non-central those amino acids that are known to be on the surface of a protein. In the figure, x-axis is the centrality and y-axis is the hydropathy index or score of an amino acid. The higher the hydropathy score of an amino acid, the more likely it is to be found in the protein core and thus is expected to be more network central. For each centrality, we identify the most network central amino acids and the least central amino acids, and then we compare their hydropathy scores. As expected, we find that the more central amino acids have high hydropathy scores and the less central amino acids have low hydropathy scores, and that there is a significant difference between the two groups. Having validated that centralities correctly capture expected positions of amino acids in a protein 3D structure, we next use them to evaluate whether the conserved rare, non-conserved rare, and common codons occupy different positions in a protein structure network. In a protein structure network, conserved rare codons can be more central than non-conserved rare codons, which we define as the relationship R1, or conserved rare codons can be less central than non-conserved rare codons, corresponding to relationship R4. Similarly, we define 4 other relationships as R2, R3, R5, and R6. We find 17 groups of proteins, where proteins within each group show the same set of relationships. This includes a group of proteins in which there is no relationship between the 3 codon categories. The figure shows the number of proteins on the y-axis that are present in a protein group on the x-axis. In 53 out of 63 considered proteins, we find significant differences between network positions of the 3 codon categories, meaning that only 10 out of the 63 proteins are present in the no relationship group. So in 53 proteins, there is some link between codon usage information and the final protein 3D structure. We demonstrate the meaningfulness of protein groups by showing that they cannot be obtained by grouping proteins based on randomized codon usage information, meaning that all of the 63 proteins now end up in the no relationship group. We further perform several additional negative control-like analyses to validate the meaningfulness of our groups. Hence, it seems that the 17 protein groups are indeed a result of a link between codon usage and the final protein 3D structure. We further explore this link by examining whether proteins in a given protein group are more similar to each other than to proteins in the other groups based on their sequences. We do not detect any differences between within group versus across group sequence similarities. However, we do find some protein groups to correlate with some secondary structural patterns, cathode domains, or biological functions. In summary, this work provides evidence that codon usage is linked to the final 3D structure of the protein, and thus possibly to the co-translational folding of the protein. Further research is needed to fully understand the meaning of these codon usage groups and investigate which of the codon usage groups might play a role in co-translational protein folding. Finally, I would like to acknowledge the support for this work by my advisor, Professor Tiana Milenkovic, my colleagues in CoLab, all of my collaborators, and the NIH funding. Thank you for listening.